Well, this is a big issue. How many of you have run into this issue where 98% similar to chimpanzees or 99%? Okay, so it's in all the churches and it's commonly taught even in colleges, Christian colleges. I really didn't know what I'd get into when I got into this area of research. I mean, I'd worked at a genomics institute and researched all kinds of critters, plants and animals and all kinds of things, but never really got into the area of human-chimp DNA similarity. It wasn't on my radar. So at the Institute for Creation Research, they asked me to investigate this area, saying, well, we're getting hit with this in all the churches. You know, we go and speak at a church, and they always ask us about this, and we need some research. So what I'm going to talk to you about today is basically the progress that I've made, and I'm still working on this area from about 4 years of research. And it's an important question because the Bible says that we're different from all the other types of creation. We're created in God's image. We're not just apes or another animal. So it's really a fundamental question, a very important question. As far as human evolution goes, the chimpanzee was selected as the main model system, supposedly our next closest relative to humans. But this hasn't really been an easy issue for evolutionists to decide. In fact, in 2009, several evolutionists published a paper saying that orangutans are our closest relatives, not chimpanzees. And they did this based on the fact that orangutans actually share more morphological or anatomical similarities to humans than chimpanzees. So this wasn't really always a cut and dry issue for scientists, but generally speaking, chimpanzee was held as kind of the majority rule decision by scientists as humans' closest relative. Not only does this not, so we have this issue of incomplete lineage sorting, or where we share some traits that are very similar between humans and chimps, but we also share some traits that are more similar anatomically between humans and orangutans. So we have this mosaic that's kind of confused evolutionists. And they saw the same thing when analyzing the DNA of chimps and orangutans and humans and gorillas. This is a quote from one paper where they say, for about 23% of our genome, we share no immediate genetic ancestry with our closest living relative, the chimpanzee. In fact, we have some parts of our genome that are more close to gorilla or more close to orangutan. So we have this mosaic going on. So it isn't this big cut and dry picture that you generally see in the popular news media. But the general consensus is that humans are at the top of the evolutionary tree, that we descended from an ancestor that we shared with chimps, and then gorilla down below that, and then orangutan down below that. But this isn't always the case when using DNA sequence or using certain anatomical traits. You can actually get things mixed up, where you have orangutan more close to humans than chimp below that, and so on. You can even have gorilla more close to humans, and so on. But there's one I want to talk to today is about genome similarity, or DNA similarity between humans and chimpanzees. So how do we determine DNA similarity? Well, there's different types of DNA similarity. So if you go to a museum or you see a show on the Discovery Channel or something, what are they talking about? Sometimes you'll hear the comment, well, we share 90% of our genes with chimpanzees, or so you'll hear that. So are you talking about genes in the genome? Or sometimes they'll be talking about protein sequences, or our protein sequences will be very similar to chimpanzee, and so on. And then also looking at images of chromosomes under microscopes with the banding patterns, and we'll talk about that towards the end of my talk when we talk about the chromosome fusion. And then DNA hybridization is, and I don't know how many scientists we have in this group that understand what DNA hybridization is, but basically you can take DNA from one organism, mix it with the DNA of another organism, and see if it comes together, if the DNA is similar enough, and we'll actually talk about that in just a few slides. But really, ultimately, the ultimate comparison between humans and chimpanzees is really with genomic sequences, or the genomes. So how did this whole human-chimp DNA similarity paradigm really get off the ground to begin with? There's actually some history to this that's important to understand. Back in the 1970s, there was a technology that was used called reassociation kinetics. This was before the era of DNA sequencing. This was before we could sequence chromosomes. And so in these studies, they would basically extract the DNA from an organism. Now, DNA is a double-stranded molecule. So what you can do is you can unzip DNA like a zipper. You know, a zipper zips up and down, and you have two parts to the zipper. Well, DNA is a double-stranded molecule. It has two parts, a plus strand and a minus strand, and they unzip. They can be separated from each other. This is called denaturing. So DNA would be denatured or unzipped with heat or with chemicals, and then it would be allowed to come back together again or zip back up. And when it comes back together again, there's different fractions that you can pull off during this process where it's reassociating or coming back together. So on this whole continuum of fast to slow or reassociation times, DNA that comes back together again or zips back up really quickly has more repeats in it. It tends to be more simple in its patterns. It has more repeat content. It's very low in genes, allegedly. And then you have low copy DNA, which is somewhere in the middle. It has a few genes in it, but still has a lot of repetitive sequence in it. So this comes back together in the middle of the continuum. And then you have DNA sequences that zip back up very slowly, and these tend to be very complex sequences that contain a lot of protein coding genes, tend to be more complex. So before scientists could sequence genomes, this was how they divvied up the genome, so to speak. And what they did was they focused primarily on this single copy or gene-rich fraction. So how do you compare then DNA between two organisms using this technology? So with humans and chimps, you would use the technology I just talked about in the previous slide. You would isolate that single copy fraction or that gene-rich fraction from the genomes, from human and chimp. You mix the DNA from human and chimp together. You denature it or unzip it. Then you let it come back together again and reassociate. In this process, you'll get some fragments where human DNA will reassociate with chimp. And then compared to the control situation, which would be just human DNA with human DNA or chimp DNA with chimp DNA, compared to that control, you can indirectly figure out how similar those DNA sequences are to each other, allegedly. So from these studies, scientists got this 98.5% similarity. So the myth was born at this point. And what's really interesting is all the DNA sequencing studies that came after that, when that technology became more advanced. It was like these DNA sequencing studies had to kind of hold to this gold standard, so to speak. And you'll see what I mean later in just a few more slides. So there's some issues with this hybridization-based data, this reassociation kinetics-based data. They were only using the gene-rich fractions of the genome, or just a small part of the genome in their comparisons. Everything else was tossed out. Probably 95% of the DNA was actually omitted in this process. The technical process is very demanding, in a lot of ways more of an art than really a science. You would need very skilled technicians to be able to do this. So there's a lot of human error involved in this. And the data is indirectly obtained. It does not actually represent real DNA sequence. It's a very indirect form to get that. But anyways, the 98.5% actually sort of surprised scientists because they thought it would be a little bit less than that, because humans and chimps are radically different. And this picture is a little deceiving because chimps actually walk on their knuckles. But the human and the chimp anatomies are radically different. So scientists were actually surprised that the DNA was that similar at that time. So the big question is, are the human and chimp genomes really 98% similar? Well, the human genome was sequenced, the first rough draft was sequenced in 2001. And there was actually two competing sectors. There was Craig Venter represented the private sector, and then this was the guy that started BioLogos, Francis Collins, who represented the public sector of the Human Genome Project. And they basically agreed to share data and to publish the results at the same time. So one group published in Nature, the other published in Science on the exact same time, and this was 2001. So this was essentially the first really detailed rough draft of the human genome. Well, the chimp genome was then published in 2005. And the chimp genome was just a rough draft. It was called a shotgun rough draft. It was just a 5-fold redundant coverage of the chimpanzee genome. Well, I published a paper in 2011 in the Answers Research Journal. This is free. You can get this online. Well, I talk about how genomes are sequenced and the technology behind it and how that's important. Because how do you think when they sequenced all these little bits of DNA from the chimpanzee genome, how do you think they sorted all that mess out? They sort, anyone? They aligned it onto the human genome as a framework, because the human genome allegedly was chimpanzee's closest ancestor. The human genome was a very complete, very thorough, framework, genomic framework. So they took the chimpanzee genome and assembled all that DNA sequence onto the human genome as a framework. So it was biased really from the beginning. And I don't have any slides showing this, but they actually also put human genes in the chimpanzee genome that they thought should have been there that weren't there. And actually the Ensemble website, which is a major a European genomics website, they actually state this on one of their web pages. We projected protein coding genes into the chimpanzee genome that weren't there, that basically we thought should have been there. So it is really amazing. So the chimpanzee genome really isn't the best product that we have to work with, but you take what you can get. So the chimpanzee genome is a highly biased genomic sequence and framework. And what's really sad is as of 2013, this paper was published, I think, in September or October 2013, where they resequenced a bunch of chimpanzee genomes, a bunch of gorilla genomes, a bunch of orangutan genomes, and they wanted to figure out what was the genetic diversity in these different types of great ape species. How do you think they assembled the genomes? Well, here's a quote, and this isn't from the paper. You have to actually dig into the materials and methods, which is supplementary information that's buried somewhere on a website to find this. They say, as the human reference is the best annotated primate genome, most analyses were performed with mappings to the human reference. And by the way, the reference they used was not the modern reference of the human genome, which is now HG19, which is actually getting ready to be updated in July to HG20. But they actually used a 2006 version of the human genome because they basically didn't want to change their framework from what they originally assembled the chimp genome onto. So they're still using the human genome as a framework to assemble all these ape genomes. They're not standing on their own merits. So what do we talk about when we talk about DNA sequence similarity? So here's just a hypothetical piece of human sequence. And DNA is formed, from the letters T, C, A, and G, representing the different bases. So if a vertical line is connecting these two letters, that means they're the same. And so this is a common way to represent a DNA, what's called a pairwise DNA alignment. So in this case, all these sequences are exactly the same. So these sequences are 100% identical or 100% similar. So if you have a base change, then and you only have a 19 out of 20 base match, then you go down to 95% similarity. But it's really not that simple. In fact, it's much more complex than that. So typically when you're blasting or querying a target sequence, or a sequence against a target genome or a database genome, this is really what you're having to deal with. You're having to deal with fragments of your sequence that you're querying, and you get these places where the bases are different, and evolutionists call those substitutions, meaning the base was substituted. All of these terms, we're kind of stuck with using. They have evolutionary connotations to them, but we have to use them because these are the terms we're given to work with. And then you have other places where you actually have an insertion of one or more bases, and these are called insertions or deletions or indels. So what do you basically include when you're trying to figure out what the DNA similarity is or identity in this case? So if you just look at substitutions, and you're looking at a 98% similarity. And many of the algorithms that are used to determine different types of selection, whether it's positive or negative and things like that, can only use this 98%, these 98% fragments. They can't use anything else with large insertions or deletions in them. But if we look at this entire region here, it's only 81% similar if we include, you know, these chunks that are present in human, absent in chimp, present in chimp, absent in human. Does that make sense? So this is really what we're dealing with. It's actually a very complex situation to actually look at genomic similarity. So when you knock out regions like that, then you can go from something at 70% down to 98%, and then you just report on the regions that you align. And so this is what makes it in the popular media. It's not the 70%, which you'll see is about what the average is. So how do evolutionists compare genes? You always hear, well, this creature has the same genes as this creature, or these genes were shared through common descent. What are they actually looking at? Well, in genes in organisms like humans and plants and animals, they're actually very complex. They're not it's just not a single. In fact, we don't even know what the boundaries are of genes anymore because transcription is interleaved and pervasive and overlapping all over the genome now. But anyways, so this would be just a hypothetical gene. It only has 4 exons. These exons are what code for protein in the genome. The human genome has about 2% of it codes for protein that we've identified right now. But it's what they don't tell you, and then you'll hear people say only 2% of the human genome is genes. Well, that's not true at all, because 20% of the human genome is these introns in between the exons. So actually, as far as the gene space in the human genome actually is closer to about 25 to 30% of the human genome actually includes protein coding gene space. But if you're just looking at the exons, it's only about 2%. So when evolutionists will compare genes, they're typically only comparing these exons sequences. But these introns, these intervening regions, are full of genes nested within the protein coding gene. They're full of all kinds of regulatory sequences that may control that gene, or they may control a neighboring gene next to it. And then you have the promoter region in front of it, which contains all sorts of regulatory elements and sequences. Then you have the 3' region behind the gene, which also contains a lot of special signals and important regulatory sequences. So genes are really complex, but when evolutionists are just comparing genes, they're usually just talking about exons, and usually only a few exons from a gene. They're not talking about the whole thing. So when a gene will basically produce a messenger RNA, these introns will be spliced out in the mature product, and you'll get a messenger RNA. And I don't want to get into it, but actually there's a lot of transcriptional complexity and alternative splicing going on in the genome to produce multiple messenger RNAs from a single gene. But this is just a very dumbed down, kind of simplified model of how things work. This isn't really how it works. It would just blow your mind if you really got into what was really going on with this. It's even more complex than scientists can understand at this point. So anyways, then of course the messenger RNA produces a protein. But what about all these other regions? I think we should be including those in our comparative gene analyses as well. Now, I actually have a project now where I'm doing that. I don't have data to show you at this point, but I'm actually comparing all the different parts of human genes to chimpanzees, so we can get a really accurate picture of what we're dealing with these genes. So how have evolutionists achieved these estimates of high similarity? They've basically omitted the non-similar regions of the genome. They've compared only highly similar segments, and we call this cherry picking, because you're just picking the best of the best, and then you're presenting that to the general public. Of course, if you read their papers, you'll see exactly what they're doing, but that doesn't make it on the 6 o'clock news. So anyways, I actually published a paper with a guy named Jerry Bergman, but anyways, I went back through the 6 major papers in the 2000s, between 2002 and 2006, the major papers that compared parts of the chimpanzee genome to human, and genes to human. And I went through all these papers to try and figure out what data they threw out and add it back into the overall calculations. And when I did this, I came up, depending on the paper, with anywhere between 65 and 86% similarity, not 96 to 98%. And so I basically, and I went through the materials and methods, and I could, because, you know, this was the area I worked in, I could see what they did and what they didn't do and what they threw out and what they left in and all that. And I figured out 65 to 86% similarity for all these major evolutionary papers, supposed evidence of 98% similarity. And I even corresponded with a very famous author on one of the first papers that was published, and he admitted all kinds of stuff to me. It was very, very enlightening. So anyways, how does the algorithm work that compares one DNA sequence against a database or a reference genome? So basically, you start off with an initial match. So you basically determine what the size of that first word that match is going to be. Is it going to be 7 bases, 11 bases, 15 bases? And then you set that parameter in the algorithm, unless you're just using the default on the web server or something. But I do my own analyses on my own servers and set my algorithm parameters myself. So anyways, so you set an initial match size, let's say that's 11 letters. And then the algorithm keeps extending out and matching progressively, base by base, outwards, until it hits a segment or a chunk that falls below the threshold that you can also set for that algorithm, that falls below that statistical threshold, and then it stops. So, and this is the BLAST-N algorithm. This is the one that's basically the most widely used. But there's certain caveats of using this algorithm. If you were just to go to the NCBI website and use their little cut and paste BLAST-N server, there are certain caveats that you need to realize if you're just going to do some kind of an analysis like that. Because I have people come up and try and check, you know, work I did that took me, you know, months to do on Linux servers, you know, running my own algorithms. They try and pick a few places in the chimp genome or whatever and stick it in this website and blast it. Oh, I got something totally different. Well, you're not doing it right. You're not using the algorithm properly. So anyways, is what you want to do, if you're comparing one segment of one genome against another genome, you want to do a one-to-one comparison. Does that make sense? In other words, you want to compare that spot in the human genome to its counterpart in the other genome if it has one. You don't want to compare bits and pieces of that sequence all over the other genome. And that's what people end up doing when they just go to the website. That would be a lot like me going to the Dallas, Texas phone book. I'm really looking for Raymond Smith, but yet all of a sudden 74 other Smiths pop up because they share some similarity because the last word is the same. Well, I'm not looking for Jason Smith. No, I want Raymond Smith, who is at this address. And so actually, this is what we're doing in the genome. We're comparing addresses from one spot in one genome. We want to find the corresponding address and the similarity for just that address in the other genome. So typically when you blast a sequence against a genome, you'll get hits all over the place. And if you do this on a web server, it'll chain all of this together and make it look more similar than it really is. So this is what we call the top hit. So this is my query sequence. Let's say it's chimp. This is a human genome. So this would be my top hit, and that obviously corresponds to the best place in the chimpanzee genome. So I actually, before I got off on complete genome-wide comparisons between chimpanzee and human, I wanted to find the best algorithm parameters I could to compare the genomes. And so there's actually a creationist guy that blasted 40,000 chimpanzee sequences against the human genome. He used the default parameters and came up with totally errant results. So I called this guy and asked him about what he did, and he wasn't very willing to talk to me. But anyways, he told me what his data set was. It was supposedly 40,000 random chimpanzee genomic sequences. So I got a hold of the data set, and then I began corresponding with the people at NCBI, the curators of the data. Well, it turned out those 40,000 sequences were were selected to be similar to human. They weren't random at all. So anyways, I took what I could get. So there's 40,000 chimp sequences from all over the chimp genome. They weren't really random at all. They were already pre-selected and packaged to be similar to human. So anyways, I used the BLAST-N algorithm. I used 30 different algorithm parameter combinations. The 40,000 chimp sequences. I compared these not just to one version of the human genome, but to 4 different versions of the human genome. And I also conducted my experiments without and with sequence masking. This is another thing. If you go to the NCBI website and you use the browser as it is, it masks all of the repetitive sequence and does not include that in the search. And so I did experiments with and without doing that, because in my opinion, every bit of the genome is important. And now the ENCODE project is showing us that literally the entire human genome is important. So I now use basically no sequence masking whatsoever. And I used 5 different statistical thresholds, 3 different word sizes, and I ended up doing 1.2 million alignments on 4 different versions of the human genome. So actually it's 4.8 million alignments. The bottom line summary, the DNA sequences that matched up, and remember these were already determined to be similar to human. So these should have been 98%, right? No, the areas that lined up were only 86 to 89% identical, and that was just for 200 bases. These sequences on average were 740 bases long. So why wasn't the entire 740 base chunk matching up? It was because the algorithm would stop because it would hit a gap. It would hit an insertion or deletion. So the DNA, I found out the DNA similarity breaks down at stretches of less than 740 bases. So in so doing, basically I realized that if I was going to compare the chimp genome in its entirety to human, I was going to have to slice up the chimpanzee genome into optimum sized fragments. So anyway, so what was going on in this experiment? So more than so I found out that more than 200 to 500 bases of chimp DNA will not contiguously match to human on average. So what's going on is that the 740 base chunks were only matching up for a fragment of that before the algorithm couldn't extend the match any further. So basically, you know, a good bit of these fragments was not even being lined up. So something had to be done. So what is the actual genome-wide similarity between humans and chimpanzees, and how can this be determined? So if you have a human chromosome, and let's say you have 740 bases matching up, so really is what's going on is that you have chunks of DNA on average that are about 86 to 89% similar, but then you have a lot of these regions that are present in chimp, absent in human, present in human, and absent in chimp. So how do we get around this kind of a situation if we're going to really figure out how similar the genomes are? How do we include all this data to do that? So what I did is I wrote software that sequentially sliced each chimp chromosome up into small pieces or subsets. And so I started out, I did a lot of preliminary experiments to find out what the optimum range would be. And of course it varied with chromosomes. So anyways, I would slice up each chimp chromosome into smaller pieces, blast those against their human chromosome counterpart. Because the chimp genome was assembled onto the human genome, I could do this because basically all of chimp chromosome 1 matches up with human chromosome 1 and so on. So I use this optimized sequence slicing to figure out what the overall genome similarity was, and I did this chromosome by chromosome for the entire genome. So my goal was to determine chimp chromosomal DNA similarity compared to human. And the good thing was is that, you know, if you're an evolutionist and then going to complain, you're going to say, well, this region was inverted on this chromosome. There was a translocation here, an inversion here, and so on. My methodology totally got around that because it sliced everything up into small pieces. So synteny or the order of these pieces on chromosomes didn't matter. So actually this was more in favor of an evolutionary outcome than a whatever, creationist one. But I also included the sequences that were present in chimp and absent in human, and in my estimates of similarity as well. The evolutionists normally don't do. They completely ignore that data. So these are all the different sub-experiments I did for all the chromosomes. So here we have chromosome 1, 2, 3, and 4, and so on. So chimps actually have 48 chromosomes, and not 46. So I combined chimp chromosomes 2A and 2B into one chromosome. Compare that to human chromosome 2, because that's how they were assembled. So anyways, the peak experiment was basically the similarity for that, the best similarity I could achieve for that chromosome. So on average, it was about 70% similar on average. And actually, my data here on the Y chromosome matched up perfectly with a paper that was published by evolutionists in 2010 on the Y chromosome. So this was the chromosome by chromosome best similarity from the experiments that I did, and it actually varied. That's why I talked about combining chromosome 2A and 2B. So the least similar chromosome was chromosome 4. It was 61.1% similar on average. The most similar was chromosome 22, which was about 77.9% similar. So of all the papers that these evolutionists have published where they focused on a single chromosome to compare to human, which one do you think they chose? 22. Chromosome 22. But when you include all of chromosome 22, it's only 78% similar. So that's a paper I published in the Answers Research Journal on that. And what's interesting is that my data actually, it wasn't anything new. It just really confirmed what other people had already discovered but didn't make the 6 o'clock news. So anyways, this was a paper. It was published by evolutionists out of the Max Planck Institute in Germany, where they actually lined up over 3 million bases of chimpanzee sequence, random sequence. And this is a quote from their paper: only about 2/3 could be unambiguously aligned to DNA sequences in humans. Well, that's close to 70%. And actually, there was a guy named Richard Buggs. He's an evolutionary geneticist, an intelligent design guy, and he actually went back and looked through the chimpanzee 2005 rough draft paper, compared it to the human genome, not with the extensive analyses that I did, but he came up with a 70% and published a short paper on it. And then there was another group out of Italy that developed their own software to compare 10,000 bases from each chimp chromosome selected, 30 base chunks selected at random to human, and they came up with about 70%. So this isn't some wild number that I just came up with. This is confirmed by other studies and other people working in the field, including evolutionists. So what constitutes the 30% difference between chimps and humans? Repeat differences, including higher order repeats, repetitive regions, transposable elements, and there's different categories of these, which are all now proving to be completely functional and critical to the genome. The subtelomere regions, or regions right behind the ends of chromosomes, are unique among humans. They're actually unique among chimps. They're unique among gorillas and orangutans. They tend to be one of the really distinct features of the genome that make each different human or ape taxonomically distinct. And then the protein coding genes, pseudogenes, which are now proving to be functional, non-coding RNA genes. So all of these things are important things that make up that 30% difference. And evolutionists have known this, but it's just not widely discussed. Here's a paper that was published in 2003 talking about insertions and deletions that occur frequently, what I was talking about, between humans and non-human primates. So they looked at chromosome 21. They looked at about 56% of it, not the whole thing. They found 57 major insertions and deletions that ranged. They ignored all the ones that were less than 200 bases, but they found over 57 that were 200 to 8,000 bases in size, these insertions and deletions, in just looking at half of that. And of course, that was one of the most similar chromosomes that I just showed you in the previous slides. And they were randomly distributed, this was interesting to me, across both regions that contained protein-coding genes and the non-genic ones. So there wasn't a preference for gene-rich regions. They were frequently found in genic intervals, or inside genes, 35%. And they totally avoided looking at 21 megabases of human chromosome 21 that were not highly similar. So studies, and there's been other studies that have been published by evolutionists talking about these huge insertions and deletions. Here's one in 2005. They found 300 base deletions common across the genome, 3,000 plus sites. Here's another evolutionary paper. Insertions and deletions, 15,000 to 250,000 bases on chromosome 16. Here's another one in 2011, 26,509 insertions and deletions, 80 to 12,000 bases in or near genes across the genome. They only examine the gene-rich areas. So this isn't news to evolutionists that are working in the field. They know what's going on. But the general public is constantly told 98%, 98%. So I may kind of just zip through these. But it's turning out that every bit of the human genome and genomes are functional. Retro elements, these transposable elements, or what you may have heard called jumping genes, are now critical to genome function. This was Richard von Sternberg worked at the, where did he work in Washington, D.C.? The Smithsonian. Got fired basically for holding to an intelligent design view of the genome. But he published this paper while he was working there with James Shapiro on how repeated elements format genome function. And it was an incredible paper listing all the different functions and functional features found in transposon. Here's one, 2009, transposable elements in gene regulation in the evolution of vertebrate genomes. Transposable elements rewire and fine-tune the transcriptome, 2013. There's just literally papers published every week now about how important transposable elements are in the function of the genome. Well, it turns out that these are also a major part constituting the difference between human and chimpanzee genomes. So this is just looking at retro elements between chimps and humans. So you can see, and this is just a broad categorization, but you can see the contents are totally different between the two. And the human genome contains a lot of very unique retro elements that aren't even found in chimpanzee. So you can see a 71% versus 56% for just the ALU element content difference between humans and chimps. 15 and 25% for the L1s, which is a long interspersed repeat. And then there's other categories as well, totally different in their contents between humans and chimps, yet fully functional in each genome. So ALU elements are particularly interesting to me because they regulate gene expression. They're found in many protein coding transcripts, many long non-coding RNA transcripts. And there's several papers, number of papers published on that. They're very important in tagging on these long tails on the ends of genes that aid in their regulation in the genome, interacting with microRNAs. They also form circular RNAs in the genome that do regulatory things. So anyways, so this is the human genome lit up for ALU elements in green. So you can see they populate all over the human genome. So I've done some studies comparing ALU elements between chimpanzees and humans. So red is chimp and human is blue. And you can see this from chromosomes 12 to 21, for this particular ALU element, ALU YA5, it isn't even present in humans, but it's all over these chimpanzee chromosomes 12 to 22, and then major differences in their content between all the other chromosomes. And evolutionists can't explain this. It's a complete mystery to them. This is another ALU element, ALU YB9, the same thing, major differences for all the chromosomes. And I've actually done this for orangutan as well, and it actually varies between orangutan and chimp. So this was a paper published by evolutionists comparing the human and the chimpanzee Y chromosomes. 53% of the human genes that are on the Y chromosome, the human Y chromosome, were totally missing on the chimpanzee Y chromosome. Gene classes, entire gene families, 33% not even found in chimpanzee. All you men ought to be really getting excited about this. You know, you've got a girlfriend or a wife that accuses you of being a monkey or something. So I mean, you look at my Y chromosome; it's totally unique. So less than 70% similarity. I love this quote. This is from this paper. Indeed, and this paper was published in a major journal. I can't remember what it was, Science or Nature. Indeed, at 6 million years of separation, the difference in MSY gene content, this is the region they sequenced of the Y chromosome that was gene-rich. The MSY gene content in chimpanzee and human is more comparable to the difference in autosomal gene content in chicken and human at 310 million years of separation. This was a paper published when they sequenced the gorilla genome, and they found that 30% of the genome in gorilla is closer to human or chimpanzee than the latter are to each other. Once again, this mosaic. And I have yet to see a paper on the gorilla Y genome because probably it was the chimp genome was, the Y chromosome was too much of a disaster. So, I know eventually they probably will do it. They'll be forced to, but at this point, in fact, that Nature paper I showed you where they resequenced all those genomes from chimps and from orangutans and from gorillas out in the wild to determine the genetic diversity that was published in 2013. What do you think they sequenced, males or females? All females. Now, why would you do that? If you want the entire genome, you want the Y chromosome in there, right? Of course, that would be bad. Then you have people like me going in the databases and digging that out. So I guess that wouldn't be good, right? So anyways, well, it turns out that the protein coding sequences are also different between humans and chimpanzees. This was a paper published showing that 1,418 of the approximately 22,000 genes at that time were completely missing in chimpanzee. And here's a quote from the paper, which is really nice. I probably just should have read that instead. Our results imply that humans and chimpanzees differ by at least 6% in their complement of genes, which stands in stark contrast to the off-sided 1.5% difference between orthologous, that just means similar in both genomes, nucleotide sequences. So this stuff is in the evolutionary literature. You just have to dig it out. This is another paper published along the same lines where they found 1,285 protein coding genes specific to human, not in chimp. So humans and chimpanzees have not only differences in the gene content, but remember genes have exons, the protein coding regions within them that are split up by introns. Well, it turns out that even the exon differences are pretty significant in between humans and chimps. In my own research, I've found that 6% of time?
Yeah, we're a couple minutes over.
Are we?
Yeah.
Okay.
Can you wrap it up in another?
I will. Yeah, I'll miss all the chromosome fusion stuff, but that's all right. Should we just take questions then? Or?
Can anybody ask a question in 2 minutes or less?
Your slide where you showed the summary of all the chromosomes you compared, was that just on those 40,000 genes that were already turned?
No, that was a separate study after that, where I compared the whole chromosomes. No, the 40,000 genomic segments, that was a preliminary study to optimize my algorithm parameters and to really, you know, that was a preliminary study to dive into the genome comparison thing. So that study basically encouraged me to go on and do the whole genome.